My name's Brandon Allgood. I'm from Numerate. We're a local Bay Area computational AI company working in this space. We're about 10 years old. We've worked with most of the big pharmaceutical companies. We also have an internal pipeline of projects, and like I mentioned, we also have been working with the DoD. So we've been kind of working at this problem of applying AI to the problem of designing drugs, and not just driving activity, but also really supporting all discovery. So driving activity in vitro as well as ADME and toxicity properties. We've done a lot of work, like I said, with the DoD, and we're kind of internally, our DNA is computer scientists and medicinal chemists. Over the last 10 years, we've developed a custom platform that's based largely on the cloud, born and bred in the cloud. We're able to, again, like I said, support all kind of all discovery all the way up to candidate. And we've been able to incorporate all medicinal chemistry data going back to the 1950s, both patent and publication. We've now, I think at this point, we've got around 6,600 off-target models that we use routinely to screen for potential issues. And like we said before, it's largely been supported through a lot of R&D effort from the DoD. So the thing I'm going to talk about today is one of our latest products that we've been working on. It's called Toxtool. And so Toxtool essentially takes all of the publicly available data and attempts what we do is we build models of off-targets and be able to screen compounds, not only individual compounds, but sets of compounds against all of these models. And again, like I was saying, we use ChemAxon tools in a number of places. So what we have in this tool is a RESTful API server that's running Java, Tomcat, and there's some Marvin and custom libraries in the back end. On the front end, we're using Node and Express, and in our client, actually embedding a lot of the ChemAxon stuff in React.js. Like I said, we're using Marvin.js, and again, yeah, we also like the JS version versus the Java version for various reasons. We're using Marvin Beans, protonation plugin group, as well as the Marcouche enumeration. And here we're actually using, as opposed to the Marcouche tool, we're actually using the Marcouche enumeration inside of Marvin.js. So this is, again, our editor. This is the kind of interface that a user would use to enter in a set of molecules or a space. You can name the job. So if your space is quite large, you might want to subsample that space if what you're trying to do is look at. So for example, a use case here would be if you're drawing in a space of compounds and you want to evaluate those against all of the off targets. What you might be asking is a particular chemotype or class of compounds, does it seem to be more promiscuous or does it seem to have more liabilities than another? And within a given space of chemistry, is there a potential gradient away from these toxicities? And so you would enter in a space, and I'll show you the results later. So again, some of these spaces can get, because they combinatorially explode, can get very large very quickly. So we allow it to either enumerate or sample the space. And then here we can, you can edit the system and then come back later. You can save jobs. You can affect the state of the jobs. So, you can, like I said, I should have actually done some movies. Normally I do this interactively, but with this system we couldn't. So, you can enter in single molecules, or you can enter in Marcouches. You can get, pretty complicated, even in just the Marvin.js editor, this space has about 100 different molecules encoded within it. And so within our system, it's, again, what we do generally is take those and store those as MRV files within our database until submission time. At submission time, we then generate molecules. We do a number of different checks on those spaces. Do they have dangling R groups? Make sure that there aren't disconnected molecule fragments. Make sure there's not any null molecules that come out of this. And once that's been validated, we then also pKa adjust those. So that you don't have to worry ahead of time about pKa adjustment when you're designing the spaces. And then once those have been submitted, then they go into a cloud backend. So with our system, we will, in order to generate the fingerprints for those molecules to be evaluated against the models, we will actually spend up to 5 minutes per molecule looking at all the low energy space and what these molecules look like in 3D. And so what ends up happening here is you submit the job. That job is then broken into molecules, and then those are actually are executed massively parallel on the cloud or within the secure environments that this is deployed. It's actually deployed on a very beefy box with a lot of cores. And then once those results are done, they're then collected and reduced and into a tree that is then presented to the users. So the results of a space look something like this, and these are pretty familiar to most chemists as they're opioids. And you can see that what our system does is then you can start to look. Now you only see the top part of this, but this actually continues down and evaluates all 6,000 and shows at the top the compounds, both the targets as well as the mechanisms of action that have been tagged as potentially high threat. Again, this is thinking a little bit more in terms of some of the DoD and off-target ways of thinking about this. You can also start to explore. We've also combined in this our reactome, so you can actually look at what pathways might be affected, not just what targets are affected. So here, for example, this is pulled up regulation of aerobic respiration, which we all know is an effect of opioids. Then if you want to look at the space in general and not individual molecules, you can actually go to our aggregate tab and then highlight a particular target, and then what that does is then highlights the molecules and showing you which ones that it expects to have a higher effect on that target or a lower effect. And in this way, you can start to explore some of the SAR of these molecules. Oh, I'm actually missing one that I thought was here. So this is where you might be able to say, okay, you can start to look at, say, this R group. If you start to add this R group, you start to lose activity against a particular target, and you can explore further. So yeah, that's kind of what we had for this system. If anybody out there is looking for a job, we're also hiring to help us continue to do this development, both from a data science perspective as well as a UI engineer perspective.
So I'll start it off. You listed both AWS and Google.
Yes.
Do you use both? Do you have a preference? Do you allow your clients to choose? How do you?
So most of our systems are used internally or with a partner. We don't sell software. We prefer AWS, and that's largely because we do massive amounts of compute. Some of our simulations can be 100,000 cores for over 2 weeks, and so we prefer AWS largely because of the price advantage of using spot instances.
Perfect. We had a lot of conversations about it yesterday, so it was just, there seemed to be a large trend and preference of AWS over Google, and
Well, Google has a number. Google has the advantage of they have a lot of dark fiber around the world. So if there are some security reasons for doing it, their AMIs come up a little faster than AWS, or their machine instances come up faster. They've recently announced a kind of spot market approach, but the overall, AWS's offerings are larger, and they're obviously 10 times bigger than Google.
Sure. Tell us a bit more about your machine learning algorithm.
I can. So we have more than one algorithm. There's many algorithms. It's not just a one-size-fits-all type of system. So we've developed algorithms for going from maybe from hit expansion, so from a bunch of tool compounds to more advanced. We also can drive activity. So at the core, right? So at the core of machine learning, you have two problems. How do you represent the information in your training data to the model and to allow it to best learn? And then how do you deal with some of the statistical issues? So one of the problems with publicly available data is it's noisy and it's biased, and there are a lot of other statistical issues there. And so how do you deal with those? So In terms of representation, we believe, we looked at the problem 10 years ago, and everybody was kind of favoring 2D fingerprints in order to build machine learning models. But my background is my PhD is in physics, and I think of binding to a target as a three-dimensional shape and electrostatic problem. And so if you're really going to start to solve the ability to make predictions, you need to actually look into 3D. This is why we'll spend up to 5 minutes a molecule actually looking at the low energy spaces. So we use a set of proprietary representation that represents the shape and electrostatics of the low energy conformations of the molecules. And we were able to, we're willing to go 5 minutes per molecule just because of the cloud. It's horizontally scalable. If we need more compute, then we just scale it. So this is not a kind of a system that you would want to be running on a laptop. This would burn your laptop out to be able to look at that. So that's in terms of representation. For the machine learning, there's a number, and this is a whole different talk, a set of patented approaches where we're able to deal with the bias and the noise, largely using ordered pairs. And again, if you look up Numerate and patents on Google, you'll see some of those patents around that. You've already got it. Okay, good. Yeah, well, I would say recovering cosmologist. Right. My PhD was in cosmology. For example, for hit expansion, what we will do is we'll build a bipartite ranking model, or effectively a classification model. And what we'll do is we will build ordered pairs out of active compounds and inactive compounds. Now you'd say, okay, well, I thought we were talking about bias, and nobody publishes inactive compounds against a target, right? But you can always fall back on the idea that HTSs generally have a hit rate of around 0.1%. So if you pull random compounds out of the medicinal chemistry literature, you can generally assume that they're inactive, at least at a 0.1% rate. So what we'll do is actually take the active compounds that we have, and then the question is, what is active? It depends on the program. Is it 10 micromolar, 1 micromolar? It depends on where you're starting. And this is generally and we will generate pairs of active compounds and assumed inactive compounds, and we will build a machine learning model that we then apply to E-molecules, for example, for very quick kind of hit finding. We might apply it to a synthesizable set. Enamine recently is now kind of promoting their 127 million synthesizable library that they claim they can get in 4 weeks at around 85%. So we then apply that to not only inform the medicinal chemists of the SAR, but also to inform the models. So we have actually developed active learning techniques so that the models in the very beginning will actually start to suggest molecules, not necessarily because the molecule is expected to be active, but that if you make and test that molecule, it will have the most information content for the next set of modeling. So once we've exhausted the purchasables and synthesizables, we then go to de novo spaces where our medicinal chemists kind of look at the SAR. They will design spaces of 1 billion compounds, 10 billion compounds. This is where we're lighting up like 100,000 cores to search that. There we actually build ranking models, which we will then use to search those. And those ranking models are, the pairs that we generate are no longer between active and inactive. They're actually between more active and less active compounds. And here, one of the ways that we deal with some of the noise, because again, we're using data from various different places with various different types of assay conditions, is that because our training is based on pairs, we won't generate pairs between two compounds we don't expect. So we talk to the biologists and say, well, what's the intrinsic noise in this assay? And they'll say, 0.5 log units. So okay, we won't generate pairs between compounds that are maybe within 0.5. So the machine learning doesn't take its time trying to get those in the right order. And then you can also use different windows between different assays. So if you're comparing you have a wholesale assay versus an enzymatic assay, you can actually use different windows, and you can still compare those and combine those data. You could, as well as combining data from patents where everybody's trying to hide what the exact activity of the molecules are by binning them. You can also throw those in. So we can throw all of these things into our ranking models. We build ranking models. We then search billions. In the beginning, I was very naive about what medicinal chemists could dream up. They can easily dream up for a given problem a space of 1 billion or 10 billion and say, the answer is likely in there. And what we'll do is search those and come back with a ranked list, make and test those. And then as the system goes forward, we will start to add our ADME models, our toxicity models to the searching to do more lead optimization. And then when, if we have enough data under a given condition, and we feel that it's robust enough to start producing regression models, we'll start producing regression models. So there's kind of a whole list of things that we do. Okay, yeah, so any databases of those kinds of compounds would be, be very useful.